To Erik Sahai from the Francis Crick for his presentation.
Well, thank you so much to Stephen and Helen for putting this together and inviting me. It's a very pleasure to be here, and I'm slightly kind of discombobulated because I've already been speaking for 3.5 minutes according to the timer. But I'll get a move on and stop making facetious remarks. So what we're really trying to tackle, I think like pretty much everyone in this room, is getting to grips with this problem as to why cancer is so hard to treat. And I guess there are various flavours of answer to this problem, but I think one of the main ones is that it changes over time, and it's hard to hit a moving target, and we've already heard Sandy and others speak to this. So with this in mind, we want to kind of address questions like, can we understand how it might evolve, and can we predict how it might evolve? And speaking to those two kind of questions, I'd like to focus in on two slightly more specific things. First is, can we determine how the tumour microenvironment evolves over time? So I'll spend the first half of my presentation talking about that. And then the second part is, can we understand how subclones compete with one another? So moving to the tumour microenvironment, what you can see kind of appearing on the screen behind me is a kind of increasingly zoomed in image of a lung adenocarcinoma. The different colours broadly reflect different cell types, and the point to get, if you're not familiar with looking at these images, is that it's actually a complex system with many, many different components. And if we kind of focus in on some of these light blue ones here are kind of typically thought to be good guys, and when they can access these kind of cells in dark blue here, such as is going on here, that's a good thing, because that means the immune cells are potentially able to engage and eliminate the tumour. But if they can't get to them, as is the case here, then that's a bad thing. And what I hope you can appreciate is this kind of single tumour actually is very heterogeneous in terms of its local regions. Just highlighting 4 of them in boxes there that you can see are quite different. And what we might want to achieve in a clinical context is to interconvert them to be more favourable in terms of their spatial organisation. So how can we understand how, in the course of natural kind of progression of the tumour without therapy, these tumour microenvironments might change into one another, because that really is the first step to understanding how we might want to achieve control over the progression of the switching of tumour microenvironments. So if one thinks about this, there are many components to the tumour microenvironment, and what are the rates at which they change? And this is just to highlight that there are a few different, kind of timescales that we might wish to think about. So, leukocytes, so these are the white blood cells. These can change, within the matter of hours. They can traffic into the tissue, they can swarm. Other cells, cancer cells, fibroblasts, endothelial cells, these typically accumulate through proliferation, which is something that happens over a timescale of a day or two. And then, in addition to all the cells, you have, all the kind of, the structural material, the scaffolding, which is the extracellular matrix. And this is largely, the most slowly changing kind of component of the tumour microenvironment. So if it's changing slowly, can we use gradual changes in the extracellular matrix as a kind of a time signature or a maturity signature of that kind of small region of tissue? And then can we string all of those together to understand how the tissue changes over time? This is just a kind of a pretty slide to highlight that we've analysed, quite a large area of number of different tumours in rather intense detail. We've extracted all the cellular information, cell states, cell positions, etc. But what we've also done using a tool that we built a few years ago is we've derived quantitative metrics of the extracellular matrix. So not just how much there is, but how it's organised. Is it in fibres? Does it branch? Does it have gaps in it? So on and so forth. And we've done this for 4 different matrix components that you can see here. And this kind of gives us, for each of those little regions of interest that we break the tumour down into, 200 micron squares, about 50 or so metrics. And then we can kind of treat this as a sort of high-dimensional space. I mean, it's not so high-dimensional compared to some data sets. And then ask, how do these things relate to one another if we kind of look at it within that space, or we do dimensionality reduction. The dimensionality reduction is just shown here, and there's a few different things that you can do. But what I hope you can appreciate is that this is looking a little bit like either a single continuum or some different continuums. Obviously, there's some caveats around exactly how you do the dimensionality reduction. but sort of encouraged by the kind of perhaps gradual transitions that might be represented here, we thought, can we take this and then apply temporal inference methods to it and ask what might be going on in these tumours as they change over time. So this is just a smaller version of the same thing, and then we've applied two different methods for inferring how these things might change over time. And we've initiated this in the regions that are most similar to the normal lung extracellular matrix, because that we thought would be the logical starting point. And you can see that these two methods give broadly concordant kind of inferences. We could also dig a little bit deeper and then ask, are there kind of different routes from here to here or here to here, and then map these out. Again, this is all inference. This is a sort of hypothesis generating mechanism. But then with this in mind, what we can then do is we can then sort of if this is the start and we track everything along these kind of temporally inferred trajectories, can we then ask how everything else in the tumour microenvironment is changing using this as our sort of timestamp or our time signature? So this is shown in the next slide. This is just looking at the collagen extracellular matrix, but we can do this for all the cell types that are in those tumours. We can do it for different sort of substates of those. So this is just some data that reassures us that we're potentially on the right track, because you can see as the prediction is that the number of endothelial cells will decrease over time as the tumour microenvironment progresses or matures, but correspondingly there will be an increase in hypoxic cancer cells, something that makes sense. There's also be an increase in exhausted CD8 T cells, again something that would be perhaps predicted to occur based on prior knowledge. Then thinking what else can we do to this? I told you that we sort of analysed large regions of the tumours. Can we then take these and map the maturity or the kind of progression of those tumour microenvironments back into the spatial map of the tumours? So this is about a couple of centimetres by 1 centimetre of that tumour, and now what you can see, the colouring represents whether the kind of those regions based on their extracellular matrix are inferred to be kind of rather early, kind of recently kind of developed or immature microenvironments, or more mature ones in the dark colour. Here you sort of see this almost kind of concentric type of pattern, suggesting that it might be growing in a kind of a radial manner, with this being the oldest area and this being the newest area. This tumour that has a more kind of complex set of kind of transitions between the different kind of states looks a bit more mixed up. This is rather early analysis. I'd be interested to kind of hear your thoughts and feedback on it. But obviously what we're able to do is then ask how this might relate to other features of those tumours, such as their immune microenvironment, or also what their genomic evolution is looking like, because we also have extensive genomic profiling for these tumours. So this is kind of the first part of what I wanted to tell you about how we're starting to be able to understand how tumours change over time and how their microenvironment changes over time. Just a couple of conclusions. I think I've already mentioned most of this, but what I'd like to sort of reiterate to you is that we think that the trajectories in the structural microenvironment can be varied, they can be inferred, they might be linear or complex, as indicated kind of here, and that we think this relates to different growth patterns in tumours, with these kind of more simple ones with the newest kind of areas around the outside growing in a radial sort of expansive manner, and then these ones where the kind of new areas and the old areas are intermingled, having a slightly different type of growth pattern. So this kind of is now the point at which I sort of transition to the second part of my presentation, which is really about how do tumours grow. I guess the simplest idea is just that they do this, but it might be that they have growing cells evenly distributed throughout, or some other types of architecture. So in the kind of second half, I'd like to address this question: how do tumours grow, and then what is the relationship between the growth pattern and evolutionary trajectories, and finishing on the point that Sandy raised so eloquently earlier, which is how does any of this relate to therapeutic outcomes? So now we'll kind of move to some of the experiments that my team does, and actually looking at tumour colonies growing by imaging them over time. So this is a couple of movies made by Sasha Bailey, and what you can see here, if you just look at the right-hand side initially, is whether or not the cells are proliferating. So green is a marker of proliferation, live cell marker, and magenta is a marker of cells that have exited the cell cycle. So you can see that this is kind of growing in that radial, very, very simple manner, and the cells are exiting the cell cycle in the center. What does this mean for how the cells are competing and what the kind of ultimate fitness of the cells is? So this is an equivalent type of experiment, but what we've done here is we've labeled a subset of the cells either red or yellow or with these kind of blue nuclei, and you can see how the cells are either proliferating or not over time. And what I'd like to draw your attention to is that some of the kind of patches of cells in the middle, they don't actually accumulate, they don't proliferate, whereas the ones around the edge do. And all we've done here is we've changed a red protein for a blue protein or a yellow protein. We haven't done anything to change the intrinsic fitness of those cells. So this tells us that in this kind of radially sort of expanding tumour, there's actually quite a strong effect of where a subclone arises or where a kind of a mutation might arise in terms of the likely long-term fate of that subclone and that mutation. And ones around the edge are favoured and ones in the centre are not favoured. So can we explore this a little bit deeper? And this is where it kind of comes to, I guess, the purpose of this whole meeting about in silico simulation of what's going on in these tumour models. So Zhao Fu, when he was working with us, kind of set up an agent-based model, not a million miles from the one that Sandy presented earlier. And within this model, it had a couple of things that we were interested in. One is the exit from the cell cycle that we observe in the center. We had experiments to suggest that this was the result of compressive stress building up in the center of those tumors. And the other thing that we were really interested in is how the ability of the cells to migrate might firstly lead to dissipation of that compressive stress. And if it does lead to dissipation of that compressive stress and a different growth pattern, then what would the consequences be for how the cells are competing with one another, and then what would the consequences be for how tumours might develop in the face of a therapy. So we wanted to kind of explore this sort of interplay between growth pattern, migratory dynamics, and the build-up of compressive stress. So these next movies kind of show you effectively the simulation, and here on the left as you're looking at it, we basically changed the colour of the cells exactly analogous to what we did in the experiment. And you can see that actually there's a really good concordance between what we observed in the experiment and what we observed in the model. I guess this is partly because we were careful about parameterizing the model. You can also see that it recapitulates this kind of outer proliferative zone that I showed you previously. So now with this in hand, we can ask a whole load of what-if questions. What if we change how adherent the cells are to one another? What if we change their migratory dynamics or the resistance that the environment provides to their migration? And in the next kind of set of movies, what we've done is we basically, in silico, made those cells undergo an epithelial to mesenchymal transition. So this is basically making them much more migratory and much less sticky to one another. And this is a feature commonly observed in aggressive tumours and therapy-resistant tumours. So now what you see, it almost looks a bit more kind of like a diffusing kind of gaseous system, and the kind of unequal subclone fate that you saw with the kind of subclones in the centre not proliferating, the ones around the outside proliferating effectively is rather lost. The subclones become much more diffuse, and there's much less suppression of proliferation in the centre, telling us that enhancing migration overrides this place of birth effect. So then, coming to the kind of key thing that we are really motivated by is how do we explore these ideas in patients? And obviously we can't make time-lapse movies in patients, we can't label cells blue and white and yellow and various colours, but what we do have, and again this is thanks to various collaborations that we have with Charlie Swanton and his team, is access to a lot of lung cancer data where there's very deep genomic characterization. So there's a lot of sequencing. This comes from the Lung Tracer X study. But how can we relate what I've shown you in terms of our experimental work and our in silico model to the sequencing data? So what we did here, and when I say we, this is the work of Zhao Fu, is instead of changing the yellow and the blue and the red colours, is basically gave each of the cells in the simulation a genome of 10,000 cells, just for simplicity. And then at various time steps, there could be a probability of mutation. Some of those mutations would be favourable, some would be unfavourable. This, I guess, was an unfavourable one. And then you could do the equivalent of regional biopsies of this in silico model and then derive the variant allele frequency profile for the cells within it in terms of their mutations. And what this was able to tell us is that actually if we play around with the migratory dynamics, so if there are high versus the kind of more epithelial context, the control context, that we get different variant allele frequency profiles as shown here, the blue line and the red line. So this is something that we can read out in the sequencing data from patients. So what was next done by Zhao was he, using his in silico simulations, trained a classifier to kind of predict whether patients from the TracerX cohort might have highly mixed tumours or might have tumours that were more analogous to our reference sort of context of being more epithelial. This is just to show that that endeavour was fairly successful. Obviously the model data clusters much more closely than the patient data, but it falls into very similar areas of parameter space. And now what we're able to do is with these kind of patient data is make predictions as to whether or not they might be more migratory in terms of the tumour dynamics and their growth patterns, or whether they might exhibit this kind of more epithelial-like, just growing around the outside type of growth pattern. And this is just to show you some of the actual sequencing variant allele frequencies that went into kind of positioning these dots in relation to the model data. So I won't take you through all the data, but it is giving us some, I think, quite exciting and reassuring concordance between our ideas from the model and our experiments and what we observed in the patients. This is just to show you in terms of growth patterns, which I introduced earlier, that one of these low mixed, one of the tumours that was inferred to be low mixed does indeed have this growth around the outside, whereas a high mixed one has much more kind of growth in the centre, providing support for our kind of model and our ideas as to how growth patterns and migration and subclonal competition might be interlinked. So just in the last minute or so, and I'm very grateful someone has reset the clock, I'd like to kind of address this question as to how this would impact the kind of response of tumours to therapy. So this is now where one of those kind of mutations that can occur in the model shown in red confers resistance to a therapy that's then applied at a particular time. And all we've done here is we haven't changed the frequency of the mutations or what the advantage is. All we've done is changed the migratory dynamics and the migratory dynamics of every single cell in that tumour. It's not just that the resistant ones become more migratory, every cell becomes more migratory. And what you can see is that actually the context of high motility means that actually the therapy-resistant clones win faster. And I think this speaks to what Sandy was talking about how kind of long or how shallow those U-shaped dips might be in the different patients. Is there any kind of evidence to back up this idea that motility affects the rate at which bad subclones win? So we've now returned to our experiments. These are not movies now, these are stills from movies, but now what we've done is the yellow cells that you can see here have resistance to a therapy that's being applied. And in this axis, what we've done is we've either turned migration down or turned migration up. And if we turn migration down, then actually the yellow subclones struggle to win. It's only if they happen to be kind of arising at the edge of one of those tumours, whereas if we turn migration up, they dominate and spread throughout the whole tumour really rather rapidly. So that's all I wanted to kind of show you in terms of data and analysis. I'll just finish with my conclusions. I hope I've been able to convince you that migratory dynamics within tumours impact how subclones compete, and that variant allele frequency profiles are a way through our kind of model which we use to relate to both experimental data and patient data, that we can actually kind of start to make predictions about whether there are high levels of migration within tumours and what the proliferation patterns might look like. I guess this speaks to the heart of this meeting, and that this in silico model provides effectively the glue or the linkage between the experimental and the clinical data, and enables us to explore ideas in both contexts. And then looking forward, I think this is sort of a possible route to having something analogous to a digital twin that's informed by mechanistic understanding that we derive from our experiments, but then using a combination of sequencing, histology, and potentially clinical imaging. I haven't spoken about it, but we did some work with Kevin Litchfield a couple of years ago, showing how clinical imaging can also give you a readout of subclone expansion, and that these things could all be integrated to upfront kind of generate a digital twin of a patient and then explore how different therapies might play out over time. So I can see my time is up. I'd just like to thank a huge number of people, funders, but most importantly the team. David, Zhao, Rob, Zoe and Sasha have really led this. Ajay Bhargav started it. Thanks to Charlie Swanton, all his team, and the patients from the TracerX cohort. So thank you for listening.
We've got time for Richard. Do you have a question?
Yeah, Eric, great talk. Just in the first part, you mentioned mainly tumours in terms of remodelling of the extracellular matrix. Obviously, because of different chronological epigenetic aging in patients, etc., and cancer often mimicking other disease states. I wondered if you'd looked at things like chronic obstructive airways disease or asthma even, where you've got abnormal lungs, and whether that's being seen also in patterns that are seen in tumours.
Yeah, good question. We're trying to get some IPF samples to kind of explore exactly what you're suggesting. Hopefully in 6 months I can answer your question.
Okay, great.
Sorry.
Thinking back to the first part of your talk, I'm wondering if you have a sense of the immune infiltration and whether you have some handle on how you could steer therapies.
Obviously, yeah, that's something we're thinking about actually together with Helen. We're doing some spatial analysis of the interrelationship of the immune cells and the extracellular matrix and the cancer cells. I think from our analysis at the moment, we can suggest an order of events. The next thing to do, I would say, is to combine it, say, with something like spatial transcriptomics, where we can then infer exactly what the kind of signaling networks and cell states are that are associated with the different points in progression and what changes as you go from immune infiltrated to immune excluded. And if you know what's changing in terms of the signaling network and the cell-cell communication, that gives you a list of targets to go after to guide your spatial patterns in a way that would be favourable.
I'm going to, in the interest of time, move forward if we can possibly look at Erik and the